So, hi, hello everyone. I am back again. Yes, the internet connection seems to work. Give me a second. Hello and welcome again to another Saturday evening live stream evening, well, only here where I am. I know that many of you are all around the world and sometimes at impossible times. Thank you that you're joining me again. When I say impossible times, I know for some of you it's early in the morning or late at night, or yeah. In any case, I would like to send greetings all around the world. What you can see here already here on my desk, these are tiny Daphnia water fleas. But that's not the only thing that I will be talking about today. The first couple of minutes, generally hello around the world. Jack Kramer is already asking a question about BX43. DIC microscope. If anyone can answer, please feel free. Hello from Vietnam, from South Coast, from North Germany, from the Netherlands. Salut al Chiui. Yes, Ankao LV. Greetings from Scotland. Okay, from the Philippines. Okay, from the UK, Slovenia, New Zealand. England, United States, Poland, very nice, and the sound and the stream are perfect. I'm very happy because of that, because it already happened that some time ago the stream unfortunately was interrupted and I had to completely restart. So what are we gonna do today? As promised, as announced in the thumbnail, and maybe some of you have already seen my recent video, I would like to put some of these Daphnia under the microscope. These are water fleas, but there are rotifers growing on it. I did make a video, but today I would like to spend a little bit more time on that. Another thing that I would like to do, and I kind of changed my plan around very short notice, I want to show you some microscope accessories which are a little bit not so good. Okay? Normally, I recommend some accessories, but today I want to basically talk about the less good ones. Some of them are really design problems. Some of them basically just don't work, or I don't know, they're defective or whatever. I'm just going to show you a whole assortment of accessories that I bought over the last couple of years, which unfortunately, let's put it nicely, underperformed. Okay, some of them are quite curious here. Then I also prepared a few other ideas here. I want to. I've got a jar of nematode worms, vinegar eels here, which need a reculturing. So I'll do that as well. I also, it depends a little bit on the available time. Look what I got here. I'm just gonna move the Daphnia out of the way. Do you recognize what this is? Yeah, it's from a sink. There's some biofilm on here, and I would like to put that also under the microscope, and I gotta keep this a little bit wet, otherwise it's gonna dry out. Okay, because if we're lucky, we're also able to see some bacteria on here. I just haven't been putting this under the microscope now for quite a few years now. That's why I thought I'll simply try this again. What I would like to do also right now is I would like to show you something else because. Somebody sent me also a 3D printed file, which I 3D printed. This is a nice little, how do you call this? A slide holder. And also one for the cover glasses, so you can put cover glasses in here. So thank you for having sent this to me. And then when I actually 3D printed this, I realized one thing. If I don't know if I'm able to make a stand somehow, then actually I can also store them horizontally somehow to allow it for drying. Yeah, because when you make permanent slides and when you dry the slides, then they should be, of course, stored horizontally. Yeah? So thank you very much for this, and I think just a couple of ideas, because what I've done in the past, I've also used some laser cutting and so on, but 3D printing, of course, also works. And I also wanted to show you I got a video also that I would like to show you. Because somebody sent me, I'm not going to say the name to keep it anonymous, but this one over here actually shows bacteria from a urinary bladder infection. Wow. What a specimen. What you see here are rod-shaped bacteria. Maybe these are some epithelial cells, and I kind of like to see them move around. And what I consider very interesting, and I think I'm going to ask again on how this has been done. This looks a little bit like oblique illumination using a color filter. Okay, so this is actually quite interesting in that sense, simply from the illumination technique. Yeah, so you see those tiny little rod-shaped bacteria. Yeah, yeah. Obviously, you don't want to have those in your body. At least not. I mean, we have them in our digestive system. That's fine there, but yeah. But of course, from a side of an infection, you don't want to have those, obviously. But I consider this imaging, how do you say, technique quite interesting here. Yeah. So this is what I will do now is very briefly. Yeah, let's have a look at those again. I'm going to, yeah, if you have any comments or any questions, please always indicate, yeah, like Ever Smaller has done at Microbehunter or at Oliver, so that I know that the comment or question is directed to me. Yes, something, some, yes, there are a couple of comments also in Esperanto. That's also one of my hobbies that I have. Yeah, and I wonder how, yeah, of Rotifers living on you, yeah. Mirror it and add a connecting wall to make it work for horizontal storage like that. Yeah, that's actually a good point. We're back to this one over here. This is actually a good point. Because I just, when I put it in, I realized this. If you can actually make a second one, a little bit, maybe, yeah, you kind of make it a little bit like a stand or horizontal, or maybe make it two-sided, yeah, or maybe also angle it. That would be a possibility as well. But that's actually something that I did not realize until I actually had this 3D printed, that systems like this can actually also be used for horizontal storage of slides until the mounting medium is dry. So very. Yeah, sometimes ideas simply appear as you go along. Okay, I will skip over many of the comments unless they are directed to me. And if you have, looks like Reinberg illumination could be, yeah. But there was also a little bit of an oblique. So I want to give you a little bit more background info on this. Again, maybe some of you have already seen the video which I uploaded just yesterday. I got this jar of Daphnia from a colleague at work. Who's basically, he's of course into aquariums, and he's breeding those Daphnia as fish food. I got 2 jars, this one over here and another one over there. And then he told me, well, those Daphnia, those water fleas look a little bit strange because they look kind of fuzzy, you know. And then when I looked at them also, I says, yeah, they're kind of fuzzy and maybe something is growing on them. And indeed, when I put one of them under the microscope a couple of days ago, I actually could see that there were lots of rotifers growing on them. I'm just going to repeat that again, even though, yes, I made the video. But I think it's still quite interesting, and I just thought that I'm going to share it with you again. Now, having those, these are, strictly speaking, according to my research at least, these are not parasites of the Daphnia, because they don't feed on the Daphnia. They're called epibionts, and this basically means they're just living on the water flea, just being carried around. Basically, they can still harm the water fleas. Why? Because yeah, they limit the movement of the water fleas, and also they are, of course, consuming food, right? So when I want to take out those water fleas here, what you do is I take a pipette and I've cut off the tip a little bit to make it a little bit larger. And now this is a little bit a question of luck, right? I'm going to move this up here. Usually what I do is kind of go in here. Yeah, and go down where there are more of them, and I simply try to suck up whatever I've got here. And there are already 2 in there. Yeah, on the bottom, there are also dead ones, right? And what I usually do is the following. Look, I kind of, now there are 2 of them on here, right? And no cover glass, and there are no more in here. Right, so if you wait a little bit until they kind of, because they're a little bit heavier sometimes, and then they go down, and then you can actually make a little drop here, and that's it. And what we do now is we directly put it under the microscope like this. What I could do is I just put it on here, and we're just going to have a look, and I hope that they also have those epibionts on them. So where is the scope view here? Here we go. Okay. So, and we always start at the lowest magnification. Of course, the arrow I have to remove. Right. Here they are. So I have to swing out the condenser. I'm going to remove this DIC, and that's basically regular bright field. And here they are, the two of these Daphnia. This one here has fewer on them. Yeah, but you can actually see over here that indeed there are some rotifers here growing on the surface of this Daphnia. There are actually not that many on this one over here, because the one that I've seen already yesterday and the day before yesterday was completely covered. Yeah, but those rotifers are quite actually interesting to look at, and yeah, I just thought, yeah, if we're just going to go up a little bit with the magnification, right? And you were probably not going to go much higher than this because otherwise yeah, there is no cover glass on here, and otherwise there is of course the danger that we're going to make the objective wet, right? This is how it looks like. And one of the interesting things that I have found out is that of course there are also dead Daphnia in the jar, but they do not have those rotifers on them. Yeah, so those rotifers really, they like to be on live Daphnia, right? Yeah. So, yeah, I wonder if they pick particular Daphnia for any specific reason. So the question is if there is some kind of a, I don't know, host, yeah, epibion specificity. Wouldn't be surprised, and I wouldn't be surprised if there are probably even several different species of rotifers on them, because I've actually seen that not all of them are the same. So I don't know if they are from the same species or just different developmental stages. I don't know. Yeah, it could also be that they are so specific that indeed there seems to be maybe even some kind of a codependency. I don't know, right? So nature is in any case quite diverse in that sense. Yeah. So this is something that I consider kind of fascinating, right? And over here as well. Yeah. And what I've also seen that, again, also in the video here, I have not seen those, is that occasionally some of those rotifers, they kind of detach or they're kind of thrown off. And I was able to actually put the individual rotifers also under high magnification. Yeah? So I think it's quite, yeah, something I have never seen before to that extent. So that's also one of the reasons why I want to share this with you. Okay. Yeah. So there is a couple of questions off topic. I see that you're using a microscope with infinity U-plan objectives. I'm using an Olympus BX53 microscope. And those microscopes are configurable, right? So this basically means if you want to buy a microscope from one of those big brands, you cannot just say, okay, I just want to buy a microscope, but they're going to ask you which objectives do you want, which features do you want, which, I don't know, yeah. You have to basically provide all of the specifications, specifically the objectives. Sometimes you also have different eyepieces to choose from, right? So the microscopes from those big brands are bought differently. Normally when you buy a low-cost, or fairly low-cost microscope, let's say directly from Amazon. Some people do that, so I'm also fine. Then normally you buy them out of the box, right? And you get a microscope, and that's basically, it comes pre-assembled, and that's what you get. And if you're not happy with it, you just return it, right? You cannot do that with those configured microscopes, because they will put it together for you, right? And usually you don't buy it out of the box, but the company, the microscope company, they will provide you an offer, and you have to tell them essentially what you want, and then they will ship you all of the parts, and then actually some technician from the company should show up at your house to put it together. Yeah. In my case, this was not the case, because I put the microscope together myself. Yeah. Yeah, here, that seems to be the dead exoskeleton of one of those. See, interestingly, there are no, how do you say, rotifers on it. Yeah. Maybe the species of rotifer evolved specifically for hitchhiking only. Okay. The point is the following. Could be. And as a matter of fact, I mean, during my microbiology training long time ago, we were told that there have been studies, DNA studies, genetic studies, and so on, of bacteria, and they discovered that the biodiversity is so extremely high in bacteria that it is assumed that every living animal species on Earth has at least is the host for one bacterial species that doesn't live anywhere else. right? Just to show you about the biodiversity. So if you're asking if they evolved specifically for hitchhiking only, of course they did that, but I'm actually taking it a step further. I wouldn't be surprised if there is some kind of even a codependency to specific water fleas, to a specific Daphnia. Yeah, so I'm actually that wouldn't surprise me, right? Kind of also shows that when the Daphnia dies and when you have this empty exoskeleton present, that then they seem to leave the exoskeleton and maybe try to find another host, right? So this actually shows that this is not just I would say coincidence, but actually they seem to be seeking out those Daphnia here. So I personally, I think this is, let's put in the filter again. I personally, that's actually the colors here nicer. Yeah, I personally consider this quite nice. You know what we could try? I don't know. This is look. I'm just going to show you something that I sometimes look. This here is the intestine, and you see that the intestine is fairly dark, right? So this basically means it's full. This means this Daphnia is quite well fed right now. And what I have already done a couple of months ago, it does take a little bit of time, but what you can do, maybe I'm going to do this because I've got the Daphnia, maybe that's something for next week, I hope I won't forget. If you take yeast cells and stain them, make them blue, and you feed them to the Daphnia, then after a couple of minutes, the intestine here will also turn blue, right? Because they're eating the yeast. But yeah. Before I return them, you know what I'm gonna do? Why not? Let's give them a little bit of food. Okay, I'm just gonna show you how this works. I need to take it off here, all right? And here again on the desk. Where is the desk? Here it is. Okay, and what I've got here is I've got a little bit of food, two different types of food. This here is a algae, compressed algae tablet. They're selling this as how do you call this? As a food supplement. So you're supposed to eat this stuff. And it's supposed to be healthy. I don't know why. You might as well eat vegetables in my view, right? I bought those basically those I think they were called spirulina algae. I bought those simply for food. They're tablets, and I'm always scratching off a little bit. And this is dry yeast. And what I'm doing is I'm just giving them a little bit, a tiny amount of these algae. And what I do is I simply take scissors and simply scratch off a little bit here. Yeah, and I think this should be enough. Let's put this again under the microscope. And the scope view, yeah, and you see here, these are the algae right now, and then you can actually also see the water movement better. And then let's make this a little smaller. Yeah. I think the movement of the water is visible much better. And then if they have not eaten anything for a longer time, you can actually see how their intestine starts to fill up over the next couple of minutes. And then yeah. And of course, if you feed them some algae, and of course also the rotifers on the surface, they are also going to feed on this, right? So there, in that sense, there is some kind of a competition going on. Okay. Yeah. So, yeah, I plan to feed this to my fairy shrimp to turn them red. Indeed, I mean, what I have done is I've taken some yeast and I added some, either was it methylene blue or crystal violet, simply to make them blue, right? And then I removed the stain, and then I added those stained yeast cells to the Daphnia, and you could actually see how their intestine over the next couple of minutes actually turned purple as they were eating those. Yeah. So that's basically here you see the rotifers are also quite happily feeding along. Difficult to see a little bit because of the rapid movement of the Daphnia. Right here, this is, by the way, that's apparently the heart of the Daphnia. Yeah, beating here in the back. Yeah, and let's turn. And sometimes the Daphnia, you can also see that there's some eggs inside the egg pouch. These ones probably don't. Yeah, I mean, I don't know. And sometimes the Daphnia, they actually the eggs hatch inside the mother, and then they actually give live birth. I was able to observe this once. Yeah, so just, what time is it? Yeah, just wanted to kind of show this to you a little bit, also a little bit as a how do you say, as a follow-up video. And And what I'm doing right now is I'm just trying to keep them alive if possible, as long as possible. Sometimes you have to add a little bit of water when the water evaporates, of course, and probably not a good idea to overfeed them either, right? And every now and then, again, as I mentioned, I check. You can check them to see if their intestines are full or not, and if they are, then you probably know that they're probably well fed. Yeah. What are those cross patterns? So that's a little bit I don't know, where's this the dead one? I don't know where it is. These seem to be simply the. So the question is, I don't know if these are actually individual cells, but it is true that there are these patterns in the exoskeleton of the Daphnia. I don't know. Let me quickly check. I don't know now where the dead one is. Can't find it anymore. Because the dead one, of course, it's not. Here's one. Here's a fragment, but it's difficult to see if we're able to see here the patterns as well. Yeah, the movement is simply too strong. So what I'm gonna do is quite simply I'm just gonna return this one. No, okay, both of them, because yeah. And where is the desk view again? And usually what I do, how do you return them? It's quite easy. You just take a little bit of liquid and you just rinse it off, and that's it. It's still stuck here. Okay. Then you know what I'm going to do now is I'm going to add a little bit more. This is a little bit too wet. I'm going to add a little bit more of this of the algae in here, so that the others also have something. Usually I just scratch it off a little bit. Okay, should be enough, and later on I'll do it with my second jar as well. We're gonna see how well this is able to keep. A couple of days ago I smelled it, and it smelled kind of foul. So this was actually a bad sign. If it starts to smell foul, then it's going somewhat anaerobic. I added a little bit of fresh water, and I stopped feeding it for a day or so, and then it seemed to have recovered now a little bit. But if it starts to smell foul, then there's some kind of weird decomposition going on, and you don't want that, right? So yeah, there seems to be also a pointy extrusion of the Daphnia at the. You mean at the tail? There are basically I don't know if this is what you're referring to, but there are different. How do you say? Species, obviously, and they differ in the shape, of course. Yeah? So, and probably maybe this is also a mix of different species. Wouldn't surprise me. Yeah. So, what I'm gonna do? Have you ever about going live healthy food source for your microbes? It prevents the problem of rotting food when slightly overfed. A live chlorella. I have not given live chlorella yet, because I have not grown live chlorella. I've got those tablets here, right? Maybe I don't need. Yeah, there's a whole bag that I basically bought once and they're sold as food supplements. But I have not. But these are obviously not alive anymore, right? They're compressed, yeah. But in order to give live chlorella algae, I'd have to grow them, and I have not done that yet. So let me quickly check. Please find part 2 of the question. I'm a little bit, I kind of skipped the question here. I'm a little bit now, I lost this here. Marie is asking, please find part 2 of question. Maybe the species of Rotifer evolved specifically for hitchhiking only. Have you got a source nearby to see if other water creatures carry these hitchhikers? No, I have not got... I get it. Okay. I don't have a source for this. However, at the same time, it doesn't surprise me. And from my research that I have done, apparently those epibionts on Daphnia are indeed not that rare. And it seems to be a problem when growing Daphnia, because the culture becomes unstable. right? So because those rotifers and the Daphnia are competing for the food, and apparently it can cause if you're not careful, the collapse of the Daphnia culture, right? So in that sense, but I don't know if any specific research has been done on the biodiversity of those epibionts. I would surprise me if it has been done, right? But then again, nature is so diverse that I wouldn't be surprised also if if there were sometimes maybe even new species, right? Because the more that you look around, the more you're probably also going to find. Yeah. So what I would like to do now is I would like to quickly change topics quite radically, because I want to talk a little bit about some microscope accessories. So it's a little bit of a last second change in program that I decided to do, because I ordered something. I was so happy. It came today. It came packaged in 3 boxes, and I would like to show it to you. And I'm going to send it back. And not only that, I'm also going to show you some other stuff that I bought over the last couple of years, which kind of didn't work out. But I'm going to show you, I've got to reach over, it's kind of large. Look what I got here. Here you can actually see it, right? There's still styrofoam around it. I don't know if any of you actually knows what this is. Okay, there's still some... And look at this. This is the bottom part. Look at this. This is what I call quality. Gee. Yeah, it's made of ceramics. Yeah. I don't mind. I don't mind the fact that it's not round and it kind of looks a little bit sloppy. It goes into this jar, what I'm going to show you. Okay. Yeah, so that is the thing. I see that there are a couple of more questions here. Don't worry. Please keep on asking questions in the chat. Don't worry. I'll try to answer them later. Okay. So question how I concentrate so many organisms. Okay. Some kind of a bath for centrifuge tubes. That would be actually, maybe this would be a, how do you say, finally a way how I could use it, because unfortunately it's useless. No, this actually goes on the bottom, and what you have over here is you have a lid. It's made of pretty nice glass. When I was still at university many years ago, we used this quite frequently. Okay, it's a glass container, right? It looks. Let me turn it from the side. This is how it looks like, right? Made of pretty solid glass. It's pretty, yeah, it looks rough here, but it's actually it's been ground smooth. And then you put a lid on top. And what you do is you put Vaseline here, and you can seal it off airtight. And then you can connect a vacuum pump to the whole thing. And this actually goes in here, right? So it's kind of, yeah, you can put your things in here. And you connect a vacuum pump here to suck out the air. Because of the Vaseline here on the side, it's going to be airtight. And you can create a glass vacuum chamber. Yes. Big, however. Okay, so this is actually, and I said, cool. And the reason is because, especially, what you're going to ask, some of you are going to ask, okay, fine, but what's the point? What does this have to do with microscopy? Well, when you're making, for example, mounting media, yeah, you have to degas them. You have to apply a vacuum to take out all of the dissolved gases to prevent air bubble formation. Yeah, so that's the reason why I kind of bought it, and it's kind of. And sometimes what you do is you do not only apply a vacuum over here, but you can also on the bottom here you put a silica gel. You know, this is this stuff that they have to keep objects dry. Sometimes when you buy clothing or shoes, they have these little bags with silica gel. You can put this in here, and this also absorbs moisture. And then you put this on here, and you apply a vacuum, either by using some kind of a hand pump or some kind of a water jet pump or some kind of an electrical pump, and you create a vacuum. And I said, cool. And then I can finally make my own mounting media and degas it and all these things, yeah? Okay, it doesn't work. I will tell you why. I'm going to show you, however, before I do that, I'm just going to show you the box that it came in. Actually, it came in 3 boxes. It was like Christmas, really. Honestly, yeah, yeah. It's the unpacking was phenomenal. Yeah, this was the inside box, and you see it was all padded out. And this box here was in a bigger box here, which was all padded out. And then this bigger box was in a yet bigger box here, which was the Amazon box. Yeah, there was some Chinese writing on here, which basically, which ChatGPT was not able to properly translate. Yeah, it says it's an eggplant made of two parts. An eggplant. I don't know. Maybe the text is not readable, right? In any case, it was a little bit like Christmas. I was unpacking the whole thing, and I was super happy. And I will just show you. Yeah, no, that's not the one here. Yeah, and basically there was a. Let me quickly check this. Maybe I'm able to find the other image here. There was another image that I kind of made. Yeah, here it is. Here it is. That's the one. That's from Amazon, right? Does it say vacuum over here? I mean, it's in German, in bad German. Doesn't matter. It says vacuum over here. It also says the vacuum over here. It was around 35 euros, right? It has 1 5-star thing, and so it was the cheapest one I could find. It was okay, fine, happy. So I'm just gonna order this, and then I unpacked it. Only to discover, and now that's basically the solution of why I'm going to send the whole thing back. Let me take out the lid. This is the disappointing part now. I want to connect a vacuum pump here, but the problem is there is no hole. It's closed. How is this possible? You see, there is no hole in here. Right? You have this thing here so that you're able to put a rubber, I don't know, seal around it and then connect some kind of a pipe or tube or hose or whatever so that essentially you can suck out the air. Why? Why? Okay? So it's totally useless, right? And I gotta send it back. Okay? So that of all, I mean, when I bought this, I said it doesn't have to be the best quality, it doesn't have to look nice. I don't care. What can go wrong? You know, this was kind of the thing that I was thinking. I mean, what can go wrong? But it was packaged nicely in 3 boxes. I don't know. Okay, you get the idea. Yeah, I know, a ceramic drill. I was also thinking of that. Yeah, but honestly, I don't have one. And I'm a little bit worried. Yeah, and I have no experience with actually drilling glass. I have to be honest with you. Yeah. So that's a little bit the thing. So maybe indeed that's maybe it's worth an experiment a little bit. Maybe I'm gonna have a look at the hardware shop and yeah. Because whether there's I mean, but this is almost a centimeter thick, right? It's a centimeter thick. So I don't know. Yeah? But in any case, the person who made this apparently didn't fully understand the significance of this here. Yeah, it needs a tube and a stopcock to control airflow. Yes, I get all of that. And basically, what I was kind of thinking is kind of put a rubber stopper with some seal and so on. I would have figured out something, right? But you see. I'm more into the microscopy business, you know, and I actually want to do stuff that's directly useful, and I don't want to spend too much time on tinkering around. But I have a few other things that I just want to share with you. Some other cool things which didn't work, which I bought. This one over here. Look what I got here. I've got, I don't know, 5, 6 different objects. I bought myself some time ago a DSLR adapter. This is actually a protective cap here. And what you have to do is just for protection, what you have to add is a so-called a T2 adapter ring, which connects to the DSLR camera. So this is basically, this works. I can connect this to the camera. So different cameras have their own adapter ring, and I would have used the one for Canon EOS. You just screw it on, and then you can connect the DSLR camera. And this goes into the eyepiece. Or the photo tube. And I don't know if you're able to see this. There's even a thing you can even turn this here. Look what happens when you turn it. There is, isn't this cool? There is a diaphragm in there. Right? There are only 2 problems with that. This diaphragm is totally pointless. It just reduces the field of view. Yeah. So it's we're not talking about anything Köhler illumination like. Forget it. Yeah. Yeah, so yeah, if I started to make it a little bit smaller, I'm already reducing the field of view. That's one thing. Also, it's kind of totally pointless. But the worst thing is when I plug it in into the into the eyepiece or into the photo tube, I have absolutely no possibility to get a clear picture because somehow the distances are wrong. It's always blurry. Yeah, there is no way how I can get a clear picture. Yeah, but it did come along with this here, where I have absolutely no idea what the function of this is. It almost looks a little bit like it's an adapter for larger eyepieces, but it still doesn't fit. And there's a sweating in here. I don't know where it goes. I have no idea what this is good for. The manufacturing quality is quite reasonable, I think. Yeah, it looks good. Yeah, but this is one of those products that has absolutely, I don't know, no function. Yeah? It's kind of so I couldn't get it to work. So it's basically I keep it as a curiosity a little bit. I think a couple of months ago or years ago, I actually in this channel here, I actually made a review video of this. Yeah, I just presented this. Maybe you're able to find it. Yeah, but it's totally pointless. What a pity. Yeah. Was not very expensive. Didn't send it back because I think I bought it over AliExpress or something like that. Yeah, so this is so that's a little bit. Here it's there's some tolerances here as well. So that's kind of one of those weird products. I've got another one here that I just want to show you, which is I find a little bit weird, which I also bought. This is this objective over here. Okay, this was a long time ago. It's a 40 times objective. It looks pretty high quality. It says PL. What does PL stand for? PL stands for positive low. It's a phase contrast objective, or it claims to be one. It was sold as a phase contrast objective, and I said, oh, okay, I'm just going to buy myself a phase contrast objective now, and then let's see if I'm able to get it to work somehow. Maybe make my own filter, phase contrast filter, and annulus, and then basically I got it. And then I opened it up here, and I looked through it, and I mean, unless you know a little bit about phase contrast microscopy, what do you notice? Yeah, in phase there's supposed to be a ring here. Yeah, it's not a phase contrast objective. I don't know what this is. Yeah, it's nothing. Yeah, that's kind of interesting. Yeah. The other interesting thing that I think, look at this here. It's 160 millimeter and 0. This means 0 means no cover glass. So this is some kind of optimized for no cover glass. Yeah. So this is this is kind of interesting of a variety of reasons, because this was actually sold as a phase contrast objective and clearly is not, because the phase ring is missing. Yeah. So it's also one of those curiosities here. Yeah, maybe it says plan. Yeah, that's what I also thought, but it was actually sold online as a phase contrast objective. Yeah, so this kind of the weird thing here, right? So yeah, I don't even know the company here. Optech, I have no idea. Yeah, so it's kind of also one of those curiosity. Yeah, yeah. I've seen Vorticella attached to water fleas. Is this the policy? Why not? Yeah. Also Vorticella also attach to surfaces. So I've also seen Vorticella before. So I wouldn't be surprised. Yeah, if it's the same thing. So that's another curiosity. Another thing that I want to show you, what I bought, and that is something that I fixed. Is this slide box over here? I bought a set of 5, and I consider it super annoying when they glue in the piece of paper. Right, it's glued in. But the funny thing was, but I got it fixed, is that I was not able to insert the slides because it was too narrow. Here, I could insert it here on the top, but not here in the middle. And the reason is because the sides here, they bent inwards. Because during the molding process, when they were making this, during the cooling, it was so distorted that it was actually too narrow on the inside. So I used a hair dryer and I kind of softened it and bent it apart again, so that now it's kind of usable. Yeah. So we're really talking about massive quality problems. Yeah, and I don't like the fact that these things are glued in any case. But that is something that I could get fixed, right? So during the manufacturing process, as the plastic cooled, it distorted to the extent that I was not even able to get the slides in. Yeah, so that's one of the quality issues as well. But you know what? These are really good for. You can actually put in your tools. Yeah, so that's actually also one of the ways that I've used it. Yeah. Does the objective work? I don't know if the objective works. This is, I think, I have to see. I don't remember if I've actually tried it out, because when I actually I've seen that actually it didn't contain the phase ring, I just said forget about it. Yeah, I'm not even gonna try it. Yeah. So this is a little bit the thing. I mean, otherwise I see no reason why it should not work. But then again, yeah, it's true that there don't seem to be a lot of lens elements in here. Yeah. And it wasn't super expensive in any case. Yeah. So let me try to close this again. So that's another thing that I just wanted to show you, and a couple of other weird things that I have. Yeah, this is another one. It's not a huge problem, but one that I almost where you have to be a little careful. This is also a slide box. Yeah, I don't like the fact that it started to corrode, but it opens up this way, right? Which is unusual for me, because I generally open it and then everything falls out, right? But I get it why they've done it like that, because the space problem, because the lid itself is much more narrow, right? Otherwise, the slide box was fine. But look what I've done here. In here again, they glued in a huge piece of paper with a list, and I ripped it out, and that was basically where the glue was, right? Super annoying. Yeah, but at least this is a functioning slide box. But you just have to be careful that it's opened a little bit in a unique way. But that was still a functional one. And I just also want to show you a few other things here. Where is the next thing? Just let me see. Ah, yeah. Where is this? Here it is. This is a regular 25 times eyepiece. Yeah. Yeah. Yeah, exactly. Yeah, such a bad design could have exactly. Look what I bought here. I think they cost around €3 a piece. I said they were so cheap. I said I'm gonna buy 2 of them. Look, these are so-called Barlow lenses, and they do magic, honestly, because what they do is they're able to double the magnification of the eyepiece. So if I've got a 25 times eyepiece here, and I put it in here, all of a sudden I have 25 times 2. That is now 50 times magnification. And you know what? Wow, I'm really going to go up. I'm going to add another one over here. And cool, now I have 50 times 2 times 2. I've got 100 times of magnification. And then when I use an objective, the one like, I don't know, 1,000 times objective, 100 times objective, I'm getting what 100 times 100 magnification is 10,000 times. Isn't this cool? Yeah. With this setup, I can convert my microscope, my optical microscope, into a microscope that's able to magnify 10,000 times. What a nonsense. Actually, the only reason why I bought it is because a couple of years ago I said, I want to try this out, right? And indeed, the image quality is not the main problem. It's so massively shaky that it's totally useless anyway, right? But I just wanted to show this to you. That's one of those, one of those totally It was actually I wanted to make a point that that essentially can go up with the magnification however much you want. You just add a €3 Barlow, and then you can go up. I don't know. Yeah, it goes up like crazy. Yeah, so that's one of those pretty pointless things here, which I thought was kind of fun, just for demos. And actually, I did translate this with ChatGPT. I took a picture, and then Google Translate also translates it, and it says auxiliary magnifying adapter 2 times. So the description actually says what it is. Yeah. So, but that's a little bit a curiosity. Yeah. So there is some kind of a as you can see a lens down here. Yeah. I don't know actually. Can you even? So that's. Let me see. Hope the lens doesn't fall out. It's actually only one lens. Yeah. It's totally. Yeah. It's a hollow tube with one lens. That's it. Yeah. Yeah. And you see. Yeah. That's how it works. Huh? makes yeah, it's a concave lens. So just wanted to now you also know why it's so cheap, because there's actually nothing to it. Yeah. So that's one of those curiosities. And I have another thing here that I consider a little bit annoying, where I said, come on, man, why do you make it like that? What I have is I've got a pipette, and that's simply sloppy manufacturing, right? And these are so-called pipette tips, right? So, and that's from, let me move this up a little bit, 1 milliliter microliter pipette, right? So you can, yeah, and then you can basically take up samples and so on, and then you press here and you kind of throw it off, and then it goes into the trash. And normally what you do is the following. You have this here, so you press it down, yeah, and then, yeah, like this, you press it in, and then you pull it out. The only problem is, now it works, but the only problem is that the diameter here is in such a way that if you press it down, it gets stuck. Yeah? So it's also not properly designed. So folks or companies are designing these things for a laboratory purpose, but there is no usability. Isn't anyone checking those products? That's what I don't get. You see, if I push it down, then it's difficult to get up, and sometimes it gets stuck. Yeah, well, I'm just saying, come on, man. It's so easy. That's what I don't understand. Is nobody testing these things? Yeah, yeah. It needs to be lower, so that's how it looks like. Yeah. That's a little bit, yeah. If it's kind of lower, then of course it kind of pushes against it. But this one actually gets stuck in here at the top. I don't get it. Yeah. And then another thing I just want to show you is, and maybe it's one of the last things. No, yeah, that's not a manufacturing problem, but a little bit of an annoyance that I discovered. Again, that's a feature, not a bug, but it doesn't work. Some of you might remember this is called the Biermann funnel, right? The Biermann funnel is for collecting nematodes and tardigrades, and you have a funnel here, and then you connect a tube. The only problem is it's pretty much all of the funnels I found have this little, how do you say, thing here to allow air to escape. So that's actually a feature, right? That you have this here, so that if you put it into a bottle, then air is able to escape here, so that it doesn't, it's able to flow better, right? But the problem is, if you connect a tube here, then it does not close air watertight anymore. And I had real issues. I had problems finding funnels of the proper size that don't have this. Yeah, that's an annoyance. So that's a little bit of a limitation. Of course, I could try to use glass funnels, but no. Yeah. So that's another thing. Another thing that I consider a little bit, which was actually I got fixed, is I bought myself some months ago. I don't know if you know what this is. This here. This is an ultrasound cleaner. Okay. And you fill it with water, and I kind of wanted to use this to clean microscope slides and other things, right? Especially if there's some dirt in here. And then I used it, and then it was always water was running out. Yeah, I was using this, and then it was all wet. I says, wow, where is it leaking? How can it be leaking? I mean, it didn't even fill it up until I found out that over here, I don't know if it's a very slight over here. Do you see this here? Yeah? There's a crack in the metal. How can this be? It's a manufacturing mistake. So the water was running out here. And then I wanted to send it back over Amazon, and then Amazon said, just throw it away. Yeah, we're going to refund you, and you throw it away. And what I have done, however, I bought myself some epoxy, and I opened it up, and I sealed it off from the outside. Yeah, so it's basically bought some metal epoxy. Yeah, stop buying cheap shit. Yeah, yes. Maybe you're saying exactly that. What? Yeah. Yeah. That's yeah. And last but not least, and that's maybe, I don't know. I bought myself 2 thermometers. Why? Because I wanted to do a little experiment, and I wanted to measure the heat, the temperature increase when microbes and microorganisms decompose, let's say, grass and other organic material. So you have basically, I wanted to simply measure the temperature, the metabolic heat, right? So this has been now on my table for the last, buy cheap, fix later, yes. So, and look, all you do is you turn it on here, I mean, what in the world? I mean, I mean, what's going on here? I mean, look at this. What's the whole point of going all the way down to the tenth of? I mean, actually, this is a really a nice textbook example. So I'm a teacher in school, and that's a nice example about the difference between accuracy and precision. Okay, look at this. I mean, the precision of a tenth of a degree, yeah, but they're totally different values. Yeah, so it means nothing. Right, the decimals mean nothing. So yeah, basically electronic tolerances, I don't know, but totally pointless, right? So I'm just saying, yeah, I mean, this can be compensated. What you can do is I can add or subtract some values, but essentially I bought those also because I wanted to do some microbe experiments. But yeah, now I have to, I don't know, do an additional subtraction step here. Not a big huge problem, but what's the whole point? You know, why have a digital thermometer with a tenth of a degree precision if the accuracy isn't there? Yeah. So so this is a little bit of just something I just wanted to share with you a little bit, a slight little rant. So why not? Okay. Yeah. So I'm gonna move on now a little bit. Let's do some actual microscopy again. What I would like to do is I would like to show you over here. Just a second. Good and the bad. I've got 2 jars with nematode worms. So I'm gonna show you the good one first. Yeah. So I got those again, also from a colleague, and he was growing those vinegar eels, nematodes as fish food. You cannot see them here because they're so small, but they're wiggling around. They're happy, and this jar, I don't know, is already over 2 years old, maybe, right? And every couple of months, I keep on, transferring it to a new one. So this one is fine, but you also want to keep 2 jars. Why? Because look at the other one. Something went really bad here. Yeah, yeah, it grew all moldy. Ooh, should not be the case. So I'm going to dump this out here, and I can also see that the how do you say, the number of nematodes actually are also decreased. The fungus is growing on the surface, so maybe this also limits the oxygen exchange. Yeah, so this can also be a problem, I guess. But what I want to do is I want to try reculture the healthy culture into a new one, and that's yeah, and then we're also gonna look at this under the microscope. Now, a few or one personal comment here. There are a couple, not a lot, maybe 1 or 2 channels out there on YouTube. They put stuff under the microscope, I don't know, some food under the microscope, and then what do you see? Nematode worms. Oh my gosh, how disgusting and how horrible. No. Yeah, that's not the case, right? I don't know. They were probably just showing these samples here as well. So I know that many of you have probably already seen those before because I keep on returning to them every now and then. I'm just for the sake of observation, I'm just going to put a small sample here under the microscope again, and then I'm going to reculture them into a new jar, which is pretty easy to do. But this also has to be done because otherwise, yeah, maybe this culture is also going to collapse sooner or later. So I have, I'm gonna take those pipettes here, yeah, these here, yeah. And the diameter is of course big enough, and all you do is you take, and I'm gonna take one from the very bottom. Okay, the rest goes back. Do nematodes have specific adaptations that allow them to live in vinegar since it's slightly acidic? It's very acidic. And these ones, yes, have, I mean, there are different types of nematodes, obviously, right? But these ones, yes, do have them. And now you're going to say, why would they actually live in vinegar? How is this? Where in the world, in nature, would you find vinegar? And actually, it's like this, that they can be found in decomposing fruits. So when apples or other fruits fall to the ground and then basically they start to decompose, then bacteria will ferment the sugar first into alcohol, and then other bacteria will convert them to vinegar, to acid. So it's a several stage process. and so it's acidic, and then those nematode worms, at least these specific types, can actually live in this acid. So that's kind of the natural environment for these worms, because otherwise, where in nature would you actually find vinegar, right? And those vinegar eels, as they're called, sometimes can actually also be found there for people who make their own vinegar if you're not really careful, right? And there is some contamination going on, maybe some flies or insects that kind of carry some of these eggs around or so on. Then of course this can be contaminated, right? So this is indeed an issue, but anyone who's who has got fish at home, an aquarium, sometimes they will actually you can buy those, right? Those vinegar eels can be bought as fish food, and then people culture them. So if you're interested in this online, you find a lot of information on this. Yeah. So let's just have a look at this them first. Yeah, just because they're kind of fun to look at. Yeah. Not all of them might be still yet alive, but because I took them from the bottom of the. Yeah. Yeah. Okay. Let me do this here. And they're a little bit compressed now between, yeah. Here they are. Ah, the questions already here. Was so fast. This hour passed quickly. Thank you. Here are some questions. So that's basically how they look like, right? I always consider them kind of fascinating. Yeah. Just from the pitcher plant. Wow, I think I have to talk to a botanical garden. Okay. Yeah, but so that's basically how they look like, and I think they're kind of now a little bit inhibited. I know not everyone likes those, right? But still fascinating creatures. They feed on bacteria, of course. Yeah. Yeasts and so on. And maybe they're also able to directly use the organic material from the vinegar, right? And I've been keeping them now and growing them for quite some time now, and I would like to reculture them. And I would say let's just do that, right? It's actually quite easy. Huh? So what you need is a 50-50. How do you say 50% of water to vinegar? I'm going to show you. This is pretty straightforward, really. Okay, and then maybe after a couple of months or so, we can revisit this again, and then we can see if they've actually reproduced properly. So so that is basically the water part. Yeah. So, and I've got some apple vinegar here, just from the store, the cheapest one available. And the idea is you have to have pretty much the same amount of water as vinegar, because pure vinegar is too strong. So I'm just going to estimate this. Yeah, so it's about up to here. Okay, so that's it. Yeah. And the next thing that you need to do is because vinegar, even though nutritious, there are a couple of important minerals and other nutrients missing. So what you want to add is an apple. Yeah, you add an apple. Okay, so that's kind of like the natural environment, that these vinegar eels like to have in any case. And now those vinegar eels are not going to develop on their own. So what I have to do is I have to inoculate. This basically means I have to add some of these. Yeah, looks like a bad urine sample. Yeah, so what I have to do, if you've not seen this yet, you take some of these here and you just inoculate. Yeah, maybe also from the bottom a little bit. Okay, and I think that should be enough already, and then you basically wait a couple of weeks, right? You can see that there's some stuff floating around already, and that's it. Okay, so yeah, just going to put this here back here a little bit. Those vinegar eels are completely harmless. They might be, not everyone likes them because they kind of look disgusting and they're worms and so on, right? But actually they're completely harmless, right? And that's it. You just leave it loose a little bit every now and then. You open it to allow it more oxygen. Yeah. From where did the original sample come from? One of my colleagues at work. He is basically he has an aquarium, and he is growing them. And you can buy those samples from an aquarium shop. They sell them, right? Also the Daphnia. You will not believe what you're able to buy online. You can buy paramecia online. You can buy, I don't know, I've seen Rotifers being sold online. Daphnia can be bought. You can buy vinegar eels, yeah, because people who have an aquarium and so on, you can buy mosquito larvae. You can whatever you want, right? You won't believe what's available. Yeah, and then once you buy them, yeah, you can continue to breed them. Right, and what I'm gonna do now is I'm just gonna wait now a little bit, a couple of weeks maybe, and so on. Then we're gonna see if this actually turns cloudy, and whether the vinegar eels are actually also able to then, reproduce here. Yeah. So no, I leave it in there. The apple slice will be left in there, right? And as a matter of fact, I don't know if you're able to see it over here. Yeah. Yeah, there is. It starts to become a little bit transparent. There is also yeah, what was once an apple slice. Yeah. Yeah, the nematodes break it down over time. Yeah. I keep what? Do you see? You keep seeing a blue comment on the desk. What do you mean the blue comment? This one over here. This yeah. This is basically crystal violet. I was not able to remove it. Yeah, that's why I've got this thing here so that my desk actually is kept. So that's next thing. What time is it? Okay. Another thing that I would like to do is biofilm. I have not tried this for some time now. So this is actually one of those things where I don't know whether it's going to be successful or not. But some time ago I did find some interesting bacteria on here. It's always moist. It's slimy, it's disgusting. So plenty of stuff should be visible here. Again, this is a little bit one of those, how do you say, things that I like to do every now and then where I don't know what the result is. And you do is the following. All you do is the following. You take a small sample from here. Ooh, look at this. Isn't it beautiful? Yeah. Usually the bacterial concentration, and I don't know. I've even a couple of years ago, I even have seen how do you call this? Some ciliates in here. I wonder how in the world did they get in there? And I now and now I'm super organized again. I've got all of the jars and everything on here, but not. I need some water. Look at this. Let's do it like this. So, and when you hope to see bacteria, then you have to make sure that they're properly suspended. Oh, here we go. And you really try to break up any clusters that there are. Oh, let's try a little more. That's way too much, right? Let's put this away again. Again, I have no real idea what to expect. So maybe it's going to be a super big disappointment. Let me trash this. But sometimes, I mean, you've already seen my video probably on the kitchen sponge. Yeah, so that's another, one. Up. Yeah, it's not quite as flat as I hoped it to be. The reason why I'm showing this is because sometimes people say, I want to see bacteria, I want to grow my own bacteria. And I'm saying, no, you're not allowed to grow your own bacteria. It's because you're at an elevated biosafety level. Yeah. If you want to see your own bacteria, try to isolate them from cheese or from yogurt or from whatever, right? Yeah. But maybe, I don't know. Let's see how this is not. There's some clusters here of whatever. We have to go up with the magnification. Okay. This one is actually one of the more disappointing samples, I have to tell you. I think I already see that. No, there. Here we go. Maybe I'm gonna go phase contrast. And then, I was already able to see a little bit of movement. Yeah, it depends a little bit on the one in this one is from one bathroom. The other one from the shower was a little bit more interesting. I've seen. Yeah. Uh, these are pretty small actually, but what you're able to see is all of those tiny things here in the background here are all bacteria. Occasionally, you see a couple of them move around. Yeah, over here as well. But I think, yeah. There are actually quite a lot of them. I hope that you're able to see them on, yeah, over the internet, but they're not the biggest ones here. Yeah. Why am I showing? What surprises me? I'll tell you now a little bit the slightly of course bacteria. So what? How in the world are they able to survive the hot temperature? Because we're always using hot water, right? So what actually scientists, and I read this somewhere, have found out is that if you actually test those bacteria, you're going to find that many of them are, not surprisingly, thermophilic. So this means they are very heat tolerant because they're always exposed to hot water. And they've also found quite a few of thermophilic bacteria in a dishwasher, because the dishwashers are, of course, always very hot. So the biofilms that form in dishwashers essentially contain bacteria that are more heat tolerant than others. And yeah, they've actually said that might be a little bit of a hygienic problem because you cannot use high temperature to kill them off. Okay. So that's a little bit heat shock proteins, yes. I've also done a long time ago. We've been also working with heat shock proteins. Basically, you kind of heat treat them, and then this becomes active, and then it starts to, in our case, it starts to destroy the bacteria. Yeah. But essentially, what we see here is also a form of how bacteria over an evolutionary process started to adapt to the environment, right? And that would be now a nice little experiment is if I were to isolate those bacteria and if I were to see if they move, I could actually do a little test series with different temperature to see at what temperature the bacteria stop moving. That would be some kind of a, yeah? Are there any solutions? Solutions, you mean basically how you can kill them all? No, well, let's put it this way. Thermophilic bacteria are completely a normal part of our environment. You can find them also in hot springs, for example, volcanic origin. So that is, yeah. If you want to kill them off, I mean, honestly, they wouldn't be harmful really here. Not expected, well, we don't know, obviously. But what you would do is of course, if you use disinfectants or things like that, hydrogen peroxide, it pretty much kills off everything. Yeah, but you have to be a little bit also aware of one thing. Some people are becoming overly careful, overly cautious, overly panicky, and they start to disinfect everything, and that's not good. Yeah, you have to, yeah. These microbes are part of our regular normal environment. Yeah. Yeah, and I just wanted to show you that also they cannot be avoided. Yeah? I have to admit, however, yeah, look, stuff like this, what you see here? Do you see this here? This sound looks a little bit like a fungus. Yeah, these thread-like structures here. So yeah, so it's a pretty complex, I would say, a fairly complex, yeah, environment what we have here. But I also have to say I have seen biofilms from yeah, that were from the bathroom that were way more interesting than this here. Yeah. But that's also a little bit some of the, how do you say, the ad hoc experiments that I like to do during the live streams. But there are plenty of them here. So normally what you would do is if you want to figure out what are they, it's not even that difficult. You basically culture them. Most of them won't be able to grow because you're not able to recreate the ideal environment, and then you do a DNA test. Yeah? Can you see them with your Barlow? The problem, honestly, right now a camera is connected. I cannot, and I will tell you why. I will show you. Because for a simple practical reason, I cannot connect my eyepiece to this here. I'm gonna take because that's my eyepiece. It won't fit. Yeah? so that's a practical problem. Yeah. So so that's yeah. Did I miss the rotifers? There's a question here. Yes, you missed them a little bit at the start of the video. The video is gonna be available anyway, and I made a 5-minute, 6-minute video just yesterday, which contains, I would say, slightly nicer videos even than what I was able to show you today about the rotifers. Yeah, so that's pretty much it. Not super exciting, this one here, right? That it's start field. So I would say let's just how do you say, leave it as it is. What time is it now? And I'm quickly going to go through a couple of your questions, and then maybe we're gonna see. Look, stuff. I just want to show you something. Look, some people are getting overly cautious about a few things, but not cautious enough about others. This is not good. What I've got here, right? So if you want to be cautious about something, if you want to worry about something, don't worry about the nematodes, worry about this here. I will tell you why this is actually dangerous. Not dangerous, but you got to be careful. Is this, look, because I've got a high concentration of bacteria on here, and the tweezers are pointed. So if I hurt myself, right, I'm actually transferring bacteria into my body, and maybe I'm going to get an infection. So what you do in this case, obviously, is you disinfect the whole thing here with alcohol. Yeah. So I'm not concerned. You see, I'm not concerned about those bacteria here, right? So much. I'm more concerned about having them now on a place where you can actually injure you, right? Yeah. So you got to be how do you say? Some people are overly cautious about they run around disinfecting all of the surfaces because they're germaphobes, and that doesn't make sense, right? But there are cases where you have to, or you should be careful, right? If you actually hurt yourself with tweezers like this, which contain a lot of unknown bacteria, then of course you have a higher risk of getting some kind of an infection here, right? So you want to avoid that. Huh? So if you want to be afraid of stuff, then don't be afraid of those nematode worms or so, but actually of the stuff where it actually makes sense, yeah, to be a little bit hygienically cautious. Yeah? So a couple of questions, not so many today, but maybe some of you have asked more on online. Let me put this away, and then I'll go through the chat again. Some of them I might have already answered. Yeah, the BX43 DIC. Generally, my advice is the following. If you want to buy it, that's a difficult question. Generally, the question is what do you mean with experiences? You see, a lot depends on personal expectations and whether it's worth it for you, and that's the thing. And if you're able to get a used BX43 DIC somewhere, then I would say there's not a lot that can go wrong, right? Yeah. So that's, but I would say it's not a question of the microscope itself, but also what your own expectations are. Yeah. So this is something that I just wanted to mention here. I wonder how long one would have to swim around a lake to end up with an excessively large colony of rotifers living on you. It's interesting, really, because let's put it this way: I find it sometimes quite difficult to control cultures because there are so many variables. You see, sometimes when you've got a Daphnia in a jar, then some of them all of a sudden die off. Some of them start to live happily. Others grow a lot of rotifers or Vorticella on them. And I think it depends on a variety of different parameters, which might be difficult to control, right? And I don't know. I would say that you can do the math. What's the generation time of a rotifer under ideal conditions? I don't know. And then you have a doubling every couple of hours, maybe. I don't know. Yeah. So that's essentially the some comment on the on the slide box. I'm using an Olympus BX53 microscope. But as I mentioned before, As I mentioned before, that these microscopes, just the brand or model of the microscope does not say a lot because it depends how it's configured. I talked about this. So chances are pretty good that yes, that this is the case. If you feed them, I don't know that. Okay. Interesting. Okay. Yeah. So if you feed you can feed the Daphnia and so on with a different stained yeast and so on. But I don't know about this here. Yeah? Have you ever thought about growing live chlorella algae? Yeah. I've also thought about this already. No, I've not tried that as a food source. Yeah. So I've been just using those compressed tablets. Yeah. Okay. A vacuum pump. Okay. I've seen Vorticella, so it's a similar. I have a box cover slip size from 0 to 16 millimeters from objectives optimized. Does the small discrepancy make any difference? That's a good point. Okay. First of all, so when you have let me start it. That's actually almost a topic of a separate video. When you look at an objective like this one over here, you're going to see that it says 0.17. And this means that the objectives are optimized for a cover glass thickness of 0.17. However, the cover glasses have sometimes a tolerance depending also on the cost and price. Some cover glasses have a very narrow tolerance. They're all 0.17 and then they're more expensive. And some people, what they do is they use some kind of a caliper tool to actually measure it out. To use and to kind of throw away those that have the wrong thickness for high resolution work. And the question of does it make a difference is the following. It depends a lot on the numerical aperture of the objective. So basically, if you're using, let's say 100, well, with the oil, it should not make a huge difference because you're using oil anyway. But for the higher magnification objectives, it can make a difference, a small one. But the question is of whether you're actually able to notice it. Okay, so this means that what you can do is if you really want to go high resolution, you either sort through the cover glasses, or I'm going to show you now something as well. Just a second. Look. I'm going to go to my microscope again. Let's turn this down a little bit here. Okay, I'm going to show you something now. So that's you can see the how do you say? Look what this is now 20 times. I have to always adjust a little bit the. Let's have a look at the thing here. It's a little too dark. Doesn't matter. 40 times. And now look, 60 times. 60 times dry objective. It's already pretty dark. The arrow is annoying. So, and now look what I have over here. And that's something that this objective has. It has a ring here. And look down here, it says 0.17, 0. Whatever, 20 and so on. And what I can do is I can turn this ring here to adjust for the cover glass thickness for the best resolution. Okay? So what you can do is for high resolution or high numerical aperture objectives, you can actually correct for the thickness of the cover glass by turning this ring here. So it says it goes from 0.11, 14, 17, 20, 20, 0.23. Yeah. And you see that actually it does not make a huge difference. Yeah. Concerning, but maybe, so I never use this actually, but maybe for certain applications this might be relevant. Yeah. Yeah. So this is, let me quickly. It's too dark. Okay. Yeah, I'm already at the maximum brightness that I have here. Yeah? So I just wanted to, because this was the question about the cover glass thickness. Where is the? Just a second. I always have problems finding the desk here again. Buy metal ones for oil changes. They don't have the ear notch. Okay. I'm a little confused now at to what this actually refers to. Okay. Okay, maybe the ultrasound cleaner. Are these ultrasound cleaner works with cleaning slides? Okay, that's a good one. That's the reason why I bought it. I bought it for cleaning the slides because I'm going to show you what I've got here. This here is my reality. This is where all of the slides go, and they're going to dry up and they're going to be difficult for cleaning. And what I found out, however, is that actually I'm probably much faster cleaning them individually by hand than with an ultrasound cleaner. The ultrasound cleaner does work to remove the roughest, how do you say, dirt. But ultimately what I have to do or what I'm doing is, I'm going to use a clean slide now, is I'm using, indeed, I'm using hot water first to soak everything, and then under running water and then with a little bit of soap, yeah, I'm actually directly rubbing over it to remove everything. Then I rinse it, yeah, and if there are any water stains on here, calcium carbonate, then I treat that also with a little bit of vinegar, some acid. And I found this actually to be more effective. Yeah, but it might be for larger slides where there are large amounts of slides, when there's lots of stuff that's kind of, how do you say, dried in. It actually might, as a start, remove most of the dirt, especially if it's kind of baked in, yeah, so to say. Yeah, so that's but alone it's not enough. Yeah, alone just the ultrasound cleaner alone, it wouldn't be enough. Yeah. Do nematodes have specific adaptations that allow them to live in vinegar? Yeah, yes. There's so many nematodes out there. Some of them not all of them like acid, obviously, right? So the biodiversity is enormous, but those specific so-called vinegar eels are called like that because they like to be in acid. Yeah? And the natural environment are sometimes fermented fruits, yeah, that kind of fell from the tree and where decomposition started, and where bacteria started to form already acid. Yeah, so that's kind of the thing. So I'm going to go through a little bit some of the questions here. With your bare hands, glass could be sharp and you could injure yourself. Very true. Very true. Very true. And indeed, if you have a whole bunch of these things here, right? So, and for this reason, what I do is I always boil the whole thing with boiling water, and that's also something I've deliberately done, is those glass slides here that I have are of better quality, right? So they are polished on the side here, so the edges are not quite as sharp, and they also have 45 degrees, how do you say, corners here. So indeed, how do you say, if you're careful, if you don't rub here on the side but on the center, then so far I have not injured myself with those slides. However, the other slides which I'm not using anymore so much, like those over here, this one is already pretty dusty and dirty. The cheap ones, right? Actually, if you look here, then yeah, it's the edges are much sharper. Yeah, and with those you can actually cut yourself. Yeah, no, that's indeed true, and that's also one of the reasons why, for example, in medical laboratories and so on, they are actually using those slightly higher quality slides. Also, not only because the laboratory staff, it's for safety, but also if you're using those ones with the hard edges, if you are using them in a machine, because there are these machines and that automatically process those slides, for example, slide scanners and so on, and there can also be chipping of glass, and you want to avoid that inside a machine. And for this reason, yeah. And also, I just want to show you another thing. Again, the scope view. If you look at the top here, look at the side here, where right at the side. Let me turn this right where this is a place where a lot of chipping can. If you just go too fast, right, then this can actually knock off the corner of some of the slides, and that's why it's good if they have already 45-degree cut edges. Yeah, but this is actually with this one it's kind of not so fast. But I've had some microscope that snap back very quickly, and then you can actually cause small glass fragments. Yeah. Yeah, yeah. So, but I get your point. I get your point with the injury when washing slides. Yeah, because this is indeed something where I also have to be a little careful. Yeah. Do you also clean cover glasses? Yes, I carefully try to clean them. Of course, yeah. And what I've also tried to do is yes, you can use dishwashing powder, and you can also try to put the slides into the dishwasher. You basically where you put the spoons and the forks and the knives, you can try to put in the slides, and this also works. Yeah? Starting with microscopy imaging for beginners, all the camera mount stuff. Okay, that's an interesting one. Yeah, tutorial. That's a challenge a little bit because there are so many different camera systems. Yeah, but why not? Why not? Yeah. Okay, thank you for the recommendation. It's basically Tini Coco was recommending some kind of a tutorial live stream for beginners for taking pictures, or maybe also some basic image processing. Yeah, yeah. I feel safe with the Globe brand. Yes, I've also the company Globe. Just in case you're wondering why I'm using those Globe slides. So is this because the company, without me asking them, they just sent them to me, and I was quite happy. Yeah, because these are actually they're not cheap really. However, one always has to be careful when handling glass, right? And of course, if you apply enough pressure, then you still can cut yourself. I mean, yeah. But I found them generally, yeah, to be of very high quality compared to those that are, of course, cheaper. That's why they're cheaper. Yeah, because they can actually really chip, and you actually feel it. You can actually feel it that you can actually, yeah, hurt yourself. Yeah, yeah, because they're not polished, and you can also see that it's kind of very how do you say, transparent here on the side, right? And those here they've used sandpaper or some kind of, I don't know what they used, yeah, or some kind of a tool to kind of grind it off and polish it off. Yeah, yeah. So referring to the funnel. Oh, there was a question. The metal funnels for oil changes. Now I get it. Thank you for the recommendation. Okay, this refers to the funnel. There are these metal funnels that are used for changing oils in cars, and they do not have those this funny ridge, yeah, for allowing air. Yeah. Okay. Thank you very much. Is anyone using any contrast method for species specific for idea of protozoans? Our work are just electronic scopes, but they is much expensive. I can if you want to identify protozoa, specifically ciliates, the contrast method, which I have never used, but I know that basically it's been used, is so-called the silver staining method. So what you can do is late Professor Feusner from the University of Salzburg, he was an expert on this. He was a protozoologist. And basically he has done a lot of this silver staining and has done a lot of staining on ciliates, and based on the staining patterns of the cilia, he did some classification work. My personal, I don't know, it's maybe a dangerous opinion that I have. My personal view is if you want to do anything, how should I put it? I've done a lot of biochemical analysis of bacteria when I was back at the university. Everything's moving to DNA these days. Yeah, so because yeah, DNA sequencing is so much more, in many cases, I'm not saying maybe in all cases, but in many cases so much more powerful, right? That very often staining and biochemical analysis is sometimes considered a little bit outdated. Yeah, I have to be careful of what I'm saying here, because I'm sure that they still have their areas of applications, but a lot of research is moving towards sequencing. Yeah. So that is a little bit one of the things, but also, for characterization and so on, you can, staining might still be used. So for example, gram staining and so on. So it's historically relevant and so on, but these days, if you want to do anything, you're very often directly going into DNA sequencing. You just want a basic function looking at some simple pond samples. Do you need a high-end DIC system? No, definitely not. No, seriously. Okay, folks, listen. Seriously. DIC, look, DIC is nice to have. I have it because I like the colors. I'm making YouTube videos. It's a hobby for me. It's that's why. Do you need it? most people don't have it. Okay, so because it's also a cost issue, obviously, right? So, and a DIC, I mean, this is a bad example here, but basically, you see, that's bright field. Okay, dot phase contrast, that is bright field. I'm seeing the bacteria actually better than in DIC, I just realized. Yeah, and now let's put in DIC again. Yeah, because of the blue background, it might look nicer, but the thing is that there's less contrast because between the dark bacteria and the blue background, the contrast is less. Yeah. And if you basically, you've got more light like this, and over here, the background is lighter, the bacteria is the same darkness, and therefore you can actually see them a little bit better, right? So you cannot automatically say it's always, you cannot almost say it's always better, but it's just one of the additional options that you have. So, phase contrast is better for bacteria. That's exactly. So this is phase contrast now, right? You have to open it up. Actually, here now they're actually maybe now I found actually a place. Now I found a place where they're much better visible than before. I don't know, maybe. So all of those tiny dots that you see, these are bacteria. So that's basically again from this the biofilm, right? So actually, this is way better. So over here, for example, rod-shaped bacterium. I would be generally, I would say that that bacteria, because a lot of folks want to see bacteria under the microscope. I would probably say they're probably the less interesting specimens. And now again in regular bright field. So people, what time is it? It's already 1.5 hours. I just quickly go through the comments. I've used an Olympus BX at work, but I was interested in how useful they are for amateur microscopy. Useful? I mean, honestly, you don't need... People, look, there is the saying that I read somewhere, it's not from me, it says the best microscope is the one, especially for hobby now, an amateur microscopy, the best microscope is the one that you're going to use the most often, right? So it's not the question of whether you have the most expensive equipment or the fanciest optics or whatever. Nice to have, right? But if you don't make use of it, it's not necessary, right? And I think it's always very important. Are we enjoying the things that we're seeing, you know? And if you want to make YouTube videos, then of course you probably want to have a better camera system. If you want to do photography, if you just want to do if you want to have a portable field microscope, then a small one is going to be fine. Yeah. So generally, if you want to that's my general advice that I'm saying is many people want to buy a very good microscope that's going to last a lifetime. And my suggestion is well, then if you want that, then don't start with that. Yeah, start with a fairly low cost device. then you can always upgrade if you're not happy. And I think what is really important, and maybe these are going to be my closing words again for the day, what is important is not that the microscope lasts a whole lifetime, but that the interest in the environment, in nature, in observation, in microscopy, that hopefully should last a lifetime, right? And then it doesn't matter so much which device you have, because chances are pretty good that you're going to be changing them anyway. Right, so that's a little bit my personal take on this. And one thing that we should always remember is that in the past, lots of researchers have done excellent very important scientific discoveries, right, with equipment that by far was not the quality that we can have now available just for a very low price. So a microscope nowadays that maybe may cost, I don't know, maybe a couple of €100 or dollars, just a few hundred of them, it might already be far beyond that what maybe some researchers in the 19th century could hope for, right? So you always have to keep that in perspective as well, right? So having a nice device is nice and fun, but I would say is only one aspect. Yeah. So people, I think. Yeah, I think I'm going to call it a day for today. Have you looked at the biofilm from a toilet cistern? Not yet. And I will be honest with you that if you're asking for a toilet, I wouldn't know from where to take the biofilm from. Maybe inside where the water is, right? So, yeah. Probably again lots of bacteria, or maybe no. That's the thing. This is where maybe the water container where you actually collect the water. I think there must be not looked at it yet. Yeah, I'd have to have a look there. But what I'm going to do now, oh, see, the water is already evaporating. So what I'm going to do now is I'm just going to, the valve area. Yes. Yeah. I'm going to call it a day for today. Wish you all the best. Yeah. Hope that you liked it, and see you around and hopefully next week again. I'll think again of a couple of topics. If you have any suggestions or so, please inform me. I'll try to do my best, and sometimes I change my mind very short notice and then change topics around as things come along. Okay, all the best, bye bye, and see you again. Happy microbe hunting as always. Bye.